final talk for the morning is from Kathryn Iovine, Evidence of the ECM-Sema3d interactions control skeletal regeneration in the fin.
Great. Thank you very much. Okay, well, I'd like to thank the organizers as well as to thank all of you for sticking around. Let's get through this. Okay, so my lab uses the regenerating fin as a model system to study skeletal morphogenesis, and recent work in my lab has identified interactions between extracellular matrix and growth factors in mediating this process. So I'm going to tell you about that today. I hope. Okay, so it's not advancing. There we go. All right. Okay, so I wanted to start by introducing the different tissues of the fin. So the fin is made of multiple bony fin rays, and each bony fin ray is comprised of multiple bony segments, where a segment is a region of bone flanked by joints, which you can see by the arrows on the right panel, which I can't quite point to because I can't get the mouse. So what I wanted to show you on the previous slide is that the fin grows by proliferation that occurs distally through the addition of new segments. And if you were to look at a longitudinal cross-section through a single fin ray, you would see that those proliferating cells are located in the medial mesenchyme, so in the middle portion of the mesenchyme, and the red cells that are located on the cartoon of a longitudinal cross-section. Those proliferating cells are flanked by collagen-like fibers termed actinotrichia, which serve as a substrate for bone-forming cells to align and secrete bone matrix. Another type of skeletal precursor cell located in that population of lateral mesenchymal cells are joint-forming cells, and so collectively we refer to those lateral mesenchymal cells as skeletal precursor cells. Okay, so my lab is interested in how bones grow to be the appropriate size. So we use the short fin mutant to study this. So short fin mutants have short fins due in part to short fin ray segments, which you can see here, and also reduced levels of cell proliferation, which we measure as the number of histone 3 positive cells. Okay, so mutations in the gap junction gene, connexin 43, are responsible for the short fin phenotype. Connexins make up gap junction channels, which reside in the plasma membrane, and these channels are responsible for the exchange of small molecules between the cytosol of dividing cell between the cytosol of adjacent cells. So things like ions and second messengers can move through channels, but not things like proteins or nucleic acids, right? So we have a poor understanding of how this type of direct cell-to-cell communication can influence tangible things like bone size or cell division. However, mutations in human connexin 43 cause a skeletal syndrome, and so we believe that the function of connexin 43 is conserved in the skeleton. Okay, so earlier work from my lab led to the hypothesis that connexin 43 does two things in the regenerating fin, both promoting cell proliferation or fin outgrowth and suppressing joint formation or patterning, regulating patterning. So we interpret the short segment phenotype of the short fin mutant as being caused by premature joint formation. So we see connexin 43 as coordinating the processes of fin outgrowth and skeletal patterning. We'd like to understand more tangibly how that works. So to do that, we completed a microarray analysis to identify connexin 43 dependent changes in gene expression. And I'm going to tell you about two of the genes that we've analyzed from that microarray. The first is called Sema3d, which is a secreted growth factor. The mRNA for Sema3d is expressed in the lateral skeletal precursor cells adjacent to the connexin 43 positive cells located medially. As part of this study, we identified receptors that Sema3d might use. So neuropilin-2a, which is expressed in the proliferating population of cells, and which functionally appears to be connected to cell proliferation, and plexin A3, which is expressed in the lateral skeletal precursor cells and functionally is related to joint formation. So the The model from this study was that Sema3d is produced and secreted from the lateral cells, where it can then interact with its neuropilin receptor to mediate cell proliferation, and its plexin A3 receptor to influence joint formation. Okay, a second gene I'll tell you about today is called Hapln1a, which is a link protein. So link proteins link the glycosaminoglycan hyaluronic acid with proteoglycans in the extracellular matrix, such as Acan. So link proteins, or Hapln1a, stabilizes the aggregate of hyaluronic acid and Acan. The Hapln1a mRNA is expressed throughout the medial and lateral mesenchyme of the fin, where hyaluronic acid is also found. This extracellular matrix can potentially connect the two populations of cells, the dividing cells and the skeletal precursor cells. So throughout my talk, I'll refer to the Hapln1a extracellular matrix, which refers to this complex of hyaluronic acid, Acan, and Hapln1a. Okay, so the Hapln1a extracellular matrix is simply one component of a more complex extracellular matrix that exists surrounding cells. We became interested in Hapln1a because the mouse knockout for Hapln1 leads to dwarfism and cradiofacial abnormalities, so skeletal defects, as well as perinatal lethality. And in addition, hyaluronic itself is known to be involved in many biological processes such as wound healing and regeneration and cancer. So because the Hapln1a extracellular matrix and Sema3d as a secreted growth factor are both located in a common extracellular space, we wondered if Hapln1a extracellular matrix might function with Sema3d. Okay, so to test that, we first looked for evidence of synergy between Hapln1a and Sema3d gene products. To do that, our initial studies on both Sema3d and Hapln1a demonstrated that morpholino-mediated knockdown of either gene leads to the connexin 43-dependent skeletal phenotypes of reduced regenerate length, segment length, and the number of H3P positive cells. So to look for evidence of synergy, we co-injected Hapln1a and Sema3d targeting morpholinos or control mismatched morpholinos at sub-threshold concentrations where either morpholino didn't cause an effect on its own, and looked for evidence of reduced phenotypes. So we looked at this using a percent similarity, which is simply the percentage of the ratio of the morpholino treated side over the untreated side of the same fin. So 100% similarity reflects little difference between the treated side and the untreated side, and is what we see when we co-inject mismatched morpholinos for Sema3d and Hapln1a for all three phenotypes. In contrast, for the targeting morpholinos for Sema3d and Hapln1a, we see reduced regenerate length, reduced segment length, and reduced dividing cells, right? Providing evidence of synergy and suggesting a genetic interaction between these gene products. Okay, a second way to ask if these gene products might function together is to ask if overexpression of Sema3d might rescue the Hapln1a knockdown phenotypes. To do this, we used a transgenic line that was provided to us by the Halloran lab that allows us to overexpress a Sema3d GFP fusion protein behind the heat shock promoter. So shown here, you can see that after a 1 hour heat shock, we see nice upregulation of the transgene, and in the absence of heat shock, we don't see much green protein at all. Okay. So we started by doing Hapln1a knockdown in regenerating fins, as we did for our other studies. And then we followed that Hapln1a knockdown with a 1 hour heat shock to induce the expression of the transgene. And we see in these experimental animals that we get 100% similarity, or about 100% similarity, for regenerate length and segment length, right? Demonstrating that overexpression of Sema3d, in fact, is rescuing the Hapln1a knockdown phenotypes. As controls, we also tested the transgenic line without heat shock, and we failed to see rescue. And we used transgene negative siblings treated with heat shock, and we also failed to see rescue, right? So this demonstrates that the presence of the transgene is necessary for rescue, as well as induction of the overexpression of the transgene to see rescue. Okay, So that allows us to add Hapln1a to our model pathway with Sema3d, where the Hapln1a extracellular matrix is found surrounding the cells of the proliferating compartment as well as the lateral skeletal precursor cells. So we next wanted to know, so if Sema3d is secreted into this extracellular space, is it possibly being sequestered by the Hapln1a extracellular matrix? Okay, so to test this, we looked for Sema3d protein levels following Hapln1a knockdown. So before I show you that, I wanted to let you know that when we do Hapln1a knockdown, we also see that we reduce the levels of hyaluronic acid, and we see reduced levels of Acan. So knocking down Hapln1a leads to essentially a reduction of this entire extracellular matrix. Okay, so now looking at Sema3d protein levels in control versus knockdown fins. So Sema3d is detected in blue in the mesenchyme of the extracellular space. We see that Hapln1a knockdown leads to a drastic reduction in the levels of Sema3d protein. Okay. Oops. So Sema3d, as I said, might be interacting with the entire complex, or it might primarily work through interactions with either hyaluronic acid or Acan. So to try to determine what Sema3d might be interacting with, we started by looking at individual knockdowns that would reduce either the levels of HA or the levels of Acan. So we knock down HA levels by knocking down the main enzyme that synthesizes hyaluronic acid, which is called Has2, hyaluron synthase 2, which we found to be expressed in the regenerating fin. Or we knock down the gene Acan-B, which is one of the Acan genes that's also expressed in the regenerating fin. For both knockdowns of Has2 or of Acan-B, we used 2 independent targeting morpholinos, and we also demonstrated that HA levels were reduced in the Has2 knockdown and that Acan protein levels are reduced in the Acan-B knockdown. Okay. And so for the Has2 knockdown and for the Acan-B knockdown, we find that they both reduce regenerate length and segment length. You can see here, and then in the interest of space and time, I left out the cell proliferation graphs, but cell proliferation is also reduced following Has2 knockdown and Acan-B knockdown. Okay, so that brings me to our model, which is shown here. So our initial studies on Sema3d and Hapln1a demonstrated that Sema3d and Hapln1a transcription increases with increased connexin 43 expression in the medial compartment. Hapln1a is secreted and stabilizes both hyaluronin and Acan in the extracellular matrix. Sema3d is secreted from the lateral skeletal precursor cells and is sequestered by, potentially stabilized by, one or more components of the Hapln1a extracellular matrix. So we don't understand the nature of the interaction between Sema3d and the Hapln1a extracellular matrix, but that work is ongoing. Stabilized Sema3d then has the opportunity to interact with its neuropilin receptor expressed medially and its plexin A3 receptor expressed laterally in order to coordinate fin outgrowth with skeletal patterning. So we believe that both Sema3d and Hapln1a are functioning to coordinate the process of outgrowth and patterning. Sema3d by interacting with distinct cell surface receptors located in 2 different populations of cells, and Hapln1a by providing a common extracellular matrix that connects these 2 different populations of cells. Okay, so most of this work was done by a grad student that recently graduated, Lakshmi Govindan, and also an undergraduate, Cha. Thank you all for your attention, and I'd be happy to take questions.
Hi, very nice talk. I just wonder whether you checked the expression of Sema3d and Hapln1a in the adult fin, and or after resection of the adult fin, are their expression being activated in adult fin?
This is all in the adult regenerating fin.
Oh, all in the adult. Okay,
Yep.
Great.
Okay, thank you very much. Let's get lunch.
Thank all the speakers and the audience for your good questions.